We have it.
We have it.
Okay, 18 minutes.
Yes, 18 minutes. We'll say, please stick with 15 minutes because the transition. Anything.
Okay, I will be right there.
Joanna's going to do the questions, right? So what's happening now?
I don't understand.
What's going on? Shall we start? Shall I say something to get it going?
No, I know. I was just going to demand the question.
Okay. Shall I say something to get us going? Right, hello everybody and welcome to this session. The first parallel session 4 of the first parallel sessions. Apologies to people online. Hello and welcome to this session, Carbon Cycling and the Land Ocean Aquatic Continuum. So this is one of 4 parallel sessions taking place at the moment. We have 5 speakers, each of whom will get 15 minutes plus 3 minutes questions. If you want to ask a question, then you have to write it in the chat, and we have a team over here that's going to be monitoring those questions, and the one with the most thumbs is going to get asked. So if you want to, so we're going to go for popular questions. So I'm personally really excited about this session because there's some really interesting and exciting work being presented, and I put an abstract into this session that didn't get accepted as well. So, but you can go and see my poster outside. It's probably a good thing it didn't get accepted. Anyway, so Henry. So Henry's going to talk to us about Greenland.
Yes. All right, hello everyone. My name is Henry Henson. I'm a PhD student at Aarhus University. And today I'll talk to you about exploring the impacts of glacial meltwater on marine CO2 uptake potential and what we can learn from a case study in Young Sound. So, as I'm sure many of you are aware, our ocean carbon dioxide concentrations mirror what we see in the atmosphere. And this is because around a quarter of all of the CO2 that we emit ends up being absorbed by the ocean. Oh, hello. Now, this is not uniform throughout the globe. So we know that our polar oceans are the largest sinks for carbon dioxide. And this is because we have cold waters here and also highly productive waters where there's a lot of photosynthesis taking place. Now, if we look at the coastal ocean, we can see that it is much more heterogeneous than the rest of the world, but it does exhibit comparable or even higher uptake rates of CO2. So it is a very interesting place to study, albeit a puzzle at some times. And if we zoom in a little bit closer, we can see that Greenland and Greenland's coastal waters rank among the highest of these uptake rates in our coastal waters globally. And yet, they are still fairly uncertain, and that is due to a few different factors. Namely that we have a very heterogeneous coastline, we have low sampling density in Greenland, and we also have prohibitively harsh conditions to sample year-round, at least in a lot of locations. Now, when we think about climate change, we can also anticipate that we're going to have more glacial meltwater in the future running off from the Greenland ice sheet. And so that means that freshwater will play a larger role in the future with regards to coastal biogeochemistry. So it's a worthy goal to try to better understand the impacts of this freshwater on air-sea CO2 exchange. Alrighty, so if we look at Young Sound briefly here, Young Sound is this fjord system located in northeast Greenland. And we can use it a bit as a natural laboratory here because it forms such a nice biogeochemical gradient between the freshwater source and the open ocean, in this case, the Greenland Sea. And we can indeed see this glacier to ocean gradient in a host of parameters. If we look here on the bottom, ooh, hello, okay. We can see that there is, yes, a distinct salinity gradient between the inner fjord where we have our freshwater being exported and then out to more saline conditions in the Greenland Sea. And when we look at PCO2 conditions, we can see that there is also a gradient where we see our lowest PCO2 conditions in the inner region of the fjord and less so in the outer region of the fjord. And this is, we see the same trend year after year. So if we look a little bit deeper into the water column, it looks a little bit something like this, where we have this distinct, where do I point? Well, anyways, a distinct freshened layer in the surface of Young Sound. And at the same time, we see in this surface freshened stratified layer, we also see lower dissolved inorganic carbon conditions because our glacial meltwater is very low in DIC. But beyond this, what are the different impacts that fresh water can have on air-sea CO2 exchange? There are a host of them which we can go through briefly. So first off, we have changes to biology. So freshwater's effect on the biological system depends upon where the freshwater is entering the fjord system. So in Greenland, we have two main glacial types, or fjord types. We have those fjords with a land-terminating glacier, and fjords with a marine terminating glacier, where the glacier is in direct contact with the ocean. And in these systems, we see that there is subglacial melt as well, and this fresh water, being buoyant, plumes upwards and carries nutrients to the surface ocean. And now, along with light and these nutrients, we can stimulate a large phytoplankton bloom, which obviously is taking up some of this carbon dioxide. And beyond that, it stimulates a much more productive food web, both in the pelagic and benthic zones. Meanwhile, if we look at a land-terminating glacier, all of the fresh water that is melted each summer gets exported via a river, essentially, only into the surface ocean. So what this does is basically enhances the stratification and sort of forms a barrier between the nutrients down below and the phytoplankton at the surface. And so oftentimes in these fjord systems, we see much less primary production and also a less productive food web as well. And we can actually see this gradient along the transect in Young Sound as well. If we look at this top graph here, we can see that there is highly turbid conditions in the inner region of the fjord and much more clear conditions in the outer region of the fjord. And this turbidity is basically how murky this water is. And the reason for that is because this glacial meltwater is also carrying a lot of silt or a lot of sediment with it when it gets exported into this fjord system. And so when we look at the actual production within this fjord, we can see that there is a gradient between autotrophy and heterotrophy dependent upon this fresh water and how much it limits our light and nutrients. So in the inner region of the fjord, can use this mouse? Okay, we see that there is net heterotrophy, basically meaning that the production is outweighing, or excuse me, the respiration is outweighing the production. Meanwhile, in the outer region of the fjord, we have net autotrophy, where we have more primary production taking place. So this is one of the impacts that freshwater can have. But if we look at the actual carbon budget of Young Sound, we can see that there's an annual uptake of around 32 grams of carbon per meter squared. But the primary production, we estimate, is only around 10 grams of carbon per meter squared. So biology is not the only thing taking place in this fjord system, and it's actually not the main driver of this atmospheric CO2 uptake. So it's these sort of physical and chemical processes which are actually driving more of the CO2 uptake in the system. And so we tried to examine a bit of these physical and chemical effects by using or by conducting a mixing experiment where we mixed seawater with different types of fresh water in order to see what would happen, all while monitoring the PCO2 of this water mass. And what we can see here is that, when we follow this mixing line or this salinity gradient, so our seawater is very elevated in dissolved inorganic carbon, and our river water, or our glacial melt, is very low in DIC. But this DIC follows this nice linear trend, which we call conservative mixing. Meanwhile, if we look at PCO2, we can see that this definitely does not show conservative mixing. Instead, we see that when we mix this glacial meltwater and the seawater, there is a rapid reduction in the PCO2 and eventually actually also an increase, which is interesting. And this is the theoretical mixing relationship we see in the blue dotted line and what we actually measured in the lab here, which matches quite closely. And this is, yeah, I guess a little bit hard to wrap your head around in the beginning when you think of mixing two atmospherically equilibrated water masses. How does it form this PCO2 reduction? But it makes a little bit more sense when you think about the rest of the carbonate system. So when we're mixing these two water masses, it's not only DIC that's being diluted, but also the alkalinity as well. So we can change the buffer capacity of this water mass when we see this fresh water coming in. And so it's actually this, yeah, eventually we see, this alkalinity becoming so diluted that we lose our buffering capacity and we see this rapid pH change. And that's where we see this CO2 actually increasing again with more fresh water. Yes, okay, and along with this effect, we wanted to see the effect of this silt. We saw that it affects turbidity in our biology a lot, but does it carry any alkalinity or change any of the chemistry as well? So we removed some of the silt from the river, and we also added this silt back into milicu water or filtered water to see what would happen. Now, first off, we wanted to see, is there any alkalinity in this silt at all? Just by adding it to milicu, over around 24 hours, we could see that yes, this silt or sediment does add some alkalinity to the water mass, this fresh water, but not a ton, and especially not a ton when we think about how much is in the ocean. And so when we removed this silt or added it, we could see that yes, it changes these PCO2 relationships slightly, but again, this alkalinity addition is quite minor when compared to how much we actually see in seawater to begin with. Okay, and then finally, we wanted to see, knowing that there are these various freshwater impacts, we wanted to see if we could examine this freshwater's impact over time or see anything here. And we have a unique opportunity to do this because Young Sound is part of this Greenland ecosystem monitoring program, which has been analyzed or measured or monitored for the past 2 decades almost. And there's a host of parameters that are measured. So if we hypothesize a conceptual model of how future change might look like in this system, we could think about something like this, where with warming temperatures, we see less sea ice and more freshwater runoff, each of those having cascading effects. Like with less sea ice, we'd have more light, more primary production, and more marine CO2 uptake, more air-sea interaction, which would lead to more uptake. This mixing-driven PCO2 reduction that I was just talking about, which would also enhance marine CO2 uptake as well. However, there are some negative effects as well with this stratification and light limitation that we also saw, which negatively impact that primary production. But all in all, we would estimate that potentially with more freshwater in the system, we'd see lower PCO2 in the surface ocean and then more uptake from the atmosphere. And we can begin to look at these trends here. If we first split the fjord into a few regions, the inner fjord, outer fjord, and the middle, we can see, first off, is there a freshening trend at all? I don't know where to point. Okay, well, in this left one, we see there isn't so much trend in the inner region of the fjord with regards to changing salinity, but this is also likely more related to whether or not there's a freshwater pulse when you're measuring this system and when we go each August. But in the middle and the outer region of the fjord, there is, yes, this trend towards lower salinities. So we do see this freshening happening over time. When we look at temperature of the surface ocean, we can also see that in each region of the fjord, there is also a slight decrease in the marine surface water temperature. So if anything, that would also enhance, lower the PCO2 of the surface ocean and enhance the uptake that we would see. However, when we look at the PCO2 conditions or the trend that we have roughly 15 years worth of data collected at this point. And just to note here, this is the delta PCO2, so the gradient between the surface ocean and the atmosphere. And we hypothesized, if we understand this system correctly, that we would see more marine CO2 uptake. But if anything, we see that this gradient between the atmosphere and the ocean is, the gradient is decreasing. It's becoming less negative here. So that's a little bit confusing, but it poses us with two questions at the moment. One being that obviously we see a ton of interannual variability here. And so one is 15 years too short of a timeline in order to be able to see this trend that we might hypothesize? Or is there something missing to our conceptual model at the moment? And this is all still quite ongoing work, so hopefully we can tease this out soon as well. But just a few conclusions that we have for the time being. We can see that, yes, Young Sound functions as this biogeochemical gradient between the freshened waters from right next to the Greenland ice sheet into the open ocean. And yes, we continue to see these lowest PCO2 conditions next to this freshwater source. In this fjord system, our physical and chemical drivers appear to play a larger role in driving this air-sea exchange within this fjord system compared to biology. And we do see some trends like freshening, but we also see a lot of interannual variability when we look at the data that we have so far. And this is work that is done by a lot of people, so I'd like to thank them for their support, and also thank you all for listening.
Thank you very much. We do have some questions online, and I want to encourage everyone to log on, because that's how you ask questions, also people from the room. So please consider logging on and asking questions online. So we have one question, which is how representative is this fjord system, in your opinion?
That's a great question. So obviously every fjord that you choose is going to be different, and I did mention that in the beginning, that in Greenland we have a lot of heterogeneity from fjord to fjord. I would say that this is an example of one type of ecosystem. So this is particularly representative of maybe a fjord system that is a land-terminating fjord, where we often see the physical and chemical factors playing a larger role than biology. And it's also representative of the east coast. So we actually see quite a large difference between the east coast and the west coast of Greenland, because there's a lot more nutrients present on the west coast. So there's generally more biology taking place in that region.
Another question from Carolina is, do you have data on alkalinity in freshwater?
Yes, we do have data on the alkalinity in freshwater. It is also quite low. So I did mention that this silt can add a little bit of alkalinity to the freshwater and to the ocean eventually when it runs off. But by and large, the freshwater, this glacial melt, is very, very low in any alkalinity. So its overall effect is to dilute the surface ocean.
And great, so we have another question from David Ho. Did you look at changes in the Schmidt number?
No, we have not looked at that yet. But in another paper that I'm working on, we're examining the effect of using different gas transfer velocity parameterizations, and we can see, it's in review at the moment, but we can see that it matters a lot, or it changes our results a lot. So I imagine that it's very relevant.
Thank you very much. If we have 1 more minute for questions, we have another question. How big are the uncertainties in your data? We did see, you mentioned the interannual variability, so how certain are you in your trends, I guess?
I mean, I think that the long-term trends are probably the most uncertain at the moment, and also what I've spent the least time analyzing as well. So I'm looking forward to digging into that. But yeah, I think 15 years is maybe not enough time to dig into this, or to find all the answers completely, particularly because we see all of this variation from year to year.
Great, thank you very much.
Thanks.
And
Okay, so this do come in. There's plenty of seats down the front. Okay, so Marta? So Marta's going to talk to us now. He's going to tell us about multiple trace gases in the coastal region.
Yes.
So Marta, you have 15 minutes, and Joanna will tell you about time. And everybody, log on to the app to ask your questions and thumbs up other people's questions.
So, hi everyone, I'm Märta Brunberg and I'm a PhD student at the University of Helsinki. And I'm here to present my data called Hotspots and Hot Moments in Greenhouse Gas Fluxes in a Diverse Coastal Ecosystem. So as I guess you all know, the mitigation of climate change demands the reduction of anthropogenic greenhouse gas emissions while also removing excess carbon from the atmosphere. And the coastal ecosystem plays an important role in global carbon and nitrogen cycles, as they can sequester a large amount of organic matter through vegetation growth, resulting in sediment accumulation and uptake of atmospheric carbon dioxide. However, the contribution of nitrous oxide in these areas to the overall greenhouse gas budget remains understudied. And this is where I come into all of this. I work with concentration and fluxes of 3 different greenhouse gases: carbon dioxide, methane, and nitrous oxide, in the surface water of shallow coastal waters. And I work alongside with ecologists and try to connect my greenhouse gas data with their knowledge of their underlying habitats, bottom substrates, and carbon stocks. So the research questions I've been working with is how spatially distinct are the 3 greenhouse gas footprints, and what is the integrated effect over a whole bay, as well as can carbon dioxide, methane, and nitrous oxide concentration of fluxes be linked to habitat types, and how significant is methane and nitrous oxide to the carbon dioxide equivalent flux estimates in shallow coastal ecosystems. To answer these questions, I've used the Vegas system, or Water Equilibrator Gas Analysis System, coupled with cavity ring-down spectroscopy. The beauty with the system is that it alternates between measuring in the concentration in air and in water. So we have the concentrations from both, as well as the sea surface temperature and salinity from each individual measurement. The water intake is approximately 30 centimeters below surface, so it's really surface water. And to calculate the fluxes, I need wind speed, and it's measured in one spot at the bay and then assumed to be the same in the entire area. So the study area I work in is a 28-hectare bay at Tvermine Zoological Station in southern Finland. It's a biologically diverse ecosystem with a mix of soft and hard bottoms and without vegetation. And it's a really shallow area, approximately 8 meters. The maximum depth is approximately 8 meters. So what I did is that I did transects during 7 campaigns, starting in October 2021, running through August 2022. I did transects for 1 to 3 days per time, and each of the circles seen here is an individual measurement. And transects like these in coastal ecosystems, while looking at greenhouse gases, is a very new method and quite a unique study. So what I did with the data is I interpolated the values. Here I have the sea surface temperature, so you can follow how the surface temperature changed throughout the year. For all my maps, the median values will be displayed along with the sample location. And now I will actually talk to you about the results. First, we have the concentration. Where should I point it? No. There we go. First, we have the carbon dioxide, and the median, the gray show the median concentration, while, gray shows approximately the air concentration, but it's a big span as it varied throughout the year as was measuring. Red is then higher concentration than air, and blue is lower. It's difficult to find a spatial pattern in these ones, but you can start seeing a pattern between the north part of the bay. See if I can get it worked. Never mind. And the south part of the bay. The lowest concentrations was found in April and May, which are months associated with biological activities such as primary production. the highest concentration the highest concentrations was found in October, December, and June, which are months then associated more with organic matter degradation. Yes. And then we have the methane. They're all above atmospheric concentration, and we can start seeing a more spatial pattern than the carbon dioxide, with the high concentrations in, the low concentrations in March and May, with May being a bit special, with high concentrations in the upper bay, but the median concentration is still very low. High concentrations were found in October, December, June, and August. October, June, and August, there is this patchiness that can be connected to where organic matter accumulation and degradation occurs, while December is a bit special. It was actually more ice in the maps than you can see here, and as we were going out with the boat measuring, the ice was breaking apart. So it's actually a thin ice cover keeping the concentration in the water. Then we have nitrous oxide, and there's no real spatial pattern, only a temporal pattern with high concentration in March and April. And again, there are high primary production months in this area. And then we have the fluxes, which are calculated. I've used Wanninghoff 2014 to calculate it, where the wind speed is the absolute dominating factor, and I measured the wind speed in one area, which was the more open part of the bay, which gives a little bit special when it comes to the results. And then we have the carbon flux, CO2 fluxes. This has a similar pattern to you could see in the concentrations, while in here you have the positive flux, which is an outgassing then, in red, and the months we see that in is October, December, and June. They're relatively small medians, and it's important to note that all of the months actually have patches where it's an inwards flux. The months with negative fluxes, which is an uptake of CO2 in the water, are April, May, March, and August, where April and May have the biggest uptake. Again, it coincides with the spring bloom, so it's not that weird. Then you have methane. It's always an outflux of methane in this area. And here we have more of a spatial pattern again. Oh, sorry. In October, June, and August, there are areas with high flux, while in April and May there is an overall high flux. And the high flux in the North Bay can be excused with there is a high concentration in the water, but as the wind is not as high up there, it's a sheltered area, it might be a bit of an overestimation. And again, December, it was covered with ice, so the reason for the high flux at this point could be that it looks like a high flux, but actually the ice was keeping it in. And then we have the nitrous oxide, which is completely different from how the concentrations looks. They're very low fluxes, however, nitrous oxide is a more potent greenhouse gas, so they're still important. The highest was in April, and that's the whole bay have a high concentration. However, for the rest of it, it's very important. If you would have measured one spot, it would have given a very weird or not a representative value for the area. And to try and connect the habitats, I, together with Alf Norco, who has extensive knowledge of the area, made this habitat map representing how the different habitats of the area looks. And for example, in May, both for carbon dioxide and methane, the Fucus habitat or bladderwrack habitat can be seen in the fluxes, as well as in August in the methane fluxes. It's more unsurprisingly that the bare sediment, along with a little bit of the mixed sediment, have a high flux of methane occurring over it. To compare the greenhouse gases, I calculated CO2 equivalents. and sorry, I'll calculate it all into CO2 equivalents using sustained global warming potential, where methane is 45 times as potent as CO2, and nitrous oxide is 270 times more potent than CO2. Here they are displayed at their own wide scale, while here they're displayed at the same wide scale. So you see that the impact of the other gases kind of disappears into it. But it's still a flux of a total of an uptake of 1.7 grams per square meter when I sum up my entire values. So it's not a budget, and it's also only daytime values, so it's not a full budget. But to round up, how spatially distinct are the three greenhouse gases? Well, methane and carbon dioxide have a similar pattern to each other, but their peak influx and outflux appear at the same time, while nitrous oxide has its own spatial pattern, but peak outflux occurs when the peak influx for carbon dioxide happens. Can carbon dioxide, methane, and nitrous oxide be concentration and fluxes be linked to habitat types? Well, methane and carbon dioxide can in a few cases, while nitrous oxide can't. So it's probably more connected to pelagic processes than benthic processes. And finally, how significant is methane and nitrous oxide for the CO2 equivalent flux estimates in shallow coastal water ecosystem? Well, it varies between the month. Methane is between 2 and 50% of the total CO2 equivalence flux, while nitrous oxide is between 0.2 and 13% of the total equivalent flux. And they're not having their peak time at the time that they're influence either. So they're both still important to the CO2 equivalence fluxes in the area. So thank you all for listening.
Thank you very much, Martha, and thank you for keeping on time. We do have some questions from online, and again, if you would like to ask questions, please feel free to do this online and also reply for the presenters to the questions asked online. So we have a question from Janne Rintala. Martha, do you think the methane is released from the benthos from deeper layers, or is this biological origin, for example, bacterial production? And also, you also have lots of sedimenting material from both phytoplankton and midsummer cyanobacterial blooms. So could it be released from their degradation?
Yes, it could be released from there. Like with the method I'm using, it's hard to find out where it's coming from in the water column, but it can definitely be from different, either of those sources.
So that this is, and then there is, the question was also, do you think methane is released from benthos, from deeper layers, or biological origin?
I think it's biological origin. There is an underlying that I'm not showing isotope values that show that it's sedimentary, older methane coming up.
Of biological origin?
Yes, of biological origin.
Thank you. There is a question from Thanos. Do you have data from the EC setup in Faramin, and if yes, do they compare?
The EC setup was set up later than this study was done, and it's not my data that's from the EC tower. There is an overlap in the CO2 data for the later months, but not for methane data.
Okay, thank you. David Ho asks, studies have shown that wind speed or gas exchange relationships in the Baltic Sea are substantially different, in parentheses lower, than open ocean parameterizations like on Icos in 2014. I'm wondering why that parameterization was chosen.
Good question. It was the one that was easiest to use when we picked it. and because we've been working with it for a while with other studies that also picked the Vanning-Hoff K to calculate it. But we do see that it would probably be better to use a lake K value in these sort of areas.
Okay, thank you. And there was one more question. Hold on one second. Can you say what causes the north-south methane gradients?
In the concentration, it's where the organic matter would accumulate and degrade. With the fluxes, the change is because of the wind, like we can see it's a wind-driven effect in this area with the methane fluxes. And there's a higher wind speed in the south than in the north. So it's the concentrations are driven out higher in the south than in the north.
Okay, thank you very much. I have a question. I was wondering, you showed the variability of substrate, I guess. It seemed like that's quite important. Would you say it's important for all the greenhouse gases?
From what I see, it's important for carbon dioxide and methane, not for nitrous oxide. It seems like nitrous oxide is more connected to pelagic work than the bottom substrates.
Okay, thank you very much. I guess.
Okay, so the next one is online. So can I check, Louise? Are you there?
Yeah.
Okay, so Louise is going to talk to us about carbon increases in temperate estuaries driven by water quality. So, oh, you're behind me, right?
Yeah.
Okay, I'll stop there and welcome and thank you very much. And how are we doing the 15 minutes and 5 minutes?
I can time myself. I've got a clock in the corner.
Okay, and Joanna's going to give you a warning.
Okay. Okay, for you. Okay, so I'll start. Thank you for the acceptance of my talk. And as I said, yeah, I'm going to give a talk on the recent inorganic carbon increase in the temperate estuary, and that was driven by water quality improvement and enhanced by droughts. So to give an introduction to the topic, so in estuaries, biogeochemical and biological processes can occur within estuaries, and these can alter the DIC concentrations, the dissolved inorganic carbon concentrations, such as primary production consumes carbon dioxide, whereas respiration requires organic carbon and releases carbon dioxide. So generally, estuaries are regarded as net heterotrophic, so that means that respiration exceeds primary production. And globally, even though the area of the estuaries is small, shown here in red, the amount of carbon dioxide they release to the atmosphere can offset the amount that's uptaken by the continental shelves. And this just shows that they're important systems as part of the global carbon cycle. So also changes in the hydrological balance in the river basins will likely regulate the biological and biogeochemical processes within estuaries. As for example, such as drought conditions can lengthen the river and estuary water residence time, and this can extend the retention of carbon and nutrients and alter their cycling within estuaries during droughts. And climate predictions show that the frequency of droughts and flood events will increase across Europe. And already between 2014 and 2020, Europe was subject to severe drought conditions. With an example shown here for Germany in summer 2015, and this drought occurred across the Elbe River basin, and which likely impacted the carbon cycling in the estuary. Okay, so along with the drought in Germany, the Elbe estuary has been impacted by human pressures for over the long-term periods. The Elbe estuary, so from our previous study, which was published last year, we found that the Elbe estuary underwent drastic changes from heavy pollution in the Elbe River basin, shown here, in the 1980s and before, which was followed by the German reunification in 1990, and the increase in wastewater treatment plants, and the reduction in nutrient loads. And this resulted, and with this, the ecosystem of the estuary was designated into 3 states. And in the recent recovery state, we found that there was a significant long-term increase in the dissolved inorganic carbon in the Elbe estuary. So our research was able to extend the recovery state by 2 years due to data availability. And we wanted to determine the trend, the drivers for this DIC increase in the estuary. And then we also wanted to assess how the recent drought period altered the carbon cycling in the Elbe estuary. So to do this, to assess the changes in the DIC, DIC and the Elbe estuary data were acquired from the FTG Elbe, which is the Elbe River Basin Community, and samples were collected by a helicopter along the estuary, with the stations shown in the black dots. And river discharge data was also included from a station in the Elbe River in the red dot. And so to describe the parameters along the estuary, which I'll go into, the region is separated into 7 zones, with the upper estuary as zone 1 to 3, which encompasses the Hamburg Harbour. And then we have the middle estuary and the lower to outer estuary. And also the FGG, Elbe, DIC, and pH data were used to calculate the TA and PCO2. Okay, so to describe the DIC dynamics in the estuary, we used a Hofmüller plot of the dissolved inorganic carbon, pH, and apparent oxygen utilization from the upper estuary along to the outer estuary in zone 7. So what we found throughout this recovery state was that during late spring and summer, in the upper estuary, in late spring and summer, the apparent oxygen utilization was negative, which is the blue values. And this indicates dissolved oxygen supersaturation. And we also found that the pH was high, and this coincided with the lowest DIC concentrations, and this indicates dominating autotrophy and the consumption of DIC. And then from zone 2, which is from the Hamburg Harbour, we can see to zone 6 in the lower estuary, we see that the apparent oxygen utilization is now positive in the orange values, and the pH decrease to low values. And this indicates a shift to dominating heterotrophy in this region. And there was also in the long estuary DIC increase, which indicates the accumulation of DIC in the surface waters. So now I'm going to go into the DIC increase drivers over the long term. So in late spring and summer, what we found is that the particular organic carbon in the upper estuary, shown here in green, increased from 1997 to 2020. And we also found a decrease in the biological oxygen demand, which suggested the improvement in water quality. Also, this increase in the POC and the dominating autotrophy in the upper region indicates the upper estuary became a larger source of POC to the mid-estuary. And then in the mid-estuary and the outer estuary, we found a significant increase in the DIC, also in late spring and summer. And also, on top of this, the magnitude of the POC concentration in the upper estuary and the magnitude of the gain in the DIC concentration in the mid-estuary were equivalent. So this indicates that the amount of POC available for remineralization was sufficient to account for the DIC production by the mid-estuary. So if I can summarize, hopefully to clarify, that the increase in the upper estuary POC increased the amount of POC that was available for remineralization by the mid-estuary, and this increased the DIC production and concentration from 1997 to 2020. This also shows the estuary's efficiency as a natural filter for POC throughout the state. So, however, the DIC gain in the lower to outer estuary was significantly higher than the upper estuary POC concentration. And their concentrations are shown here for the summer. And so this indicates that other mechanisms likely supported the DIC production in these regions, with the examples given here, such as the in situ remineralization of organic carbon from the coastal region rather than the POC from the upper region. So now moving on to describe the influence of the drought on the carbon cycling in the estuary. So during the drought, blocked out in pink, the mean Elbe River discharge was on average 32% lower than the long-term mean. And also by 2020, the river discharge in May had decreased to levels usually observed in summer, shown here in blue. And as a result, the estuary water residence time likely extended by approximately 3 times in late spring during the drought. And also in the drought, the internal DIC load was significantly higher in late spring compared to the non-drought years. And so this all indicates that the low river discharge in May allowed for a longer remineralization period of the particulate organic carbon and DIC production in the mid to lower estuary.
5
But
minutes, Louise.
Sorry?
5 minutes, sorry.
Oh, yeah. Okay. Yeah, got it. And the enhanced internal DIC load in the mid region was most likely sustained by the sufficient POC loading shown in green from the upper estuary. So we calculated the annual DIC water to air fluxes and the lateral flux during the recovery state, and we used the sum of the two as the estimated total export of DIC out of the estuary. So despite the higher internal DIC load in late spring, during the drought, the annual DIC export to the coastal waters was on average 24% lower during the drought, which is shown here in the blue values. But we didn't find a significant change in the water to air CO2 flux. And so this shows that the decrease in the river discharge during a drought did have an impact on the inorganic carbon delivery to the coastal ocean. So I'd like to conclude that the long-term DIC increase in the mid-estuary was driven by an increase in the upper estuary particulate organic carbon, as this increased the amount of POC that was available for remineralization in the estuary and subsequently increased the DIC production. Then during, in the recent drought period, the mean river discharge was significantly lower and the dry season extended into late spring. And this increased the water residence time in May and permitted a longer remineralization period of POC, and this resulted in a higher internal DIC load in late spring, shown in the purple values. Even so, the annual DIC export to the coastal waters was on average 24% lower in the drought. And this shows the impact of climate-related drought on the estuary coastal carbon budget. And as climate change models predict an increase in these extreme hydrological events, the influence of the changes in river discharge on carbon cycling and export from an estuary to coast should be considered in the global carbon budgets. So I would like to thank you for your attention and just show the acknowledgements.
Thanks, Louise. We have a question online, so I would, if you have any questions, please do ask in the chat. So first, Bruno Delil asks, what are the advantages of collecting data or samples with a helicopter?
That was time. So then all of the samples were collected during the ebb phase of the tide. And so the helicopter is faster. And it's the Elbe River Basin community who do this.
And does that have any influence on the sampling?
Well, it ensures that the samples were collected during the same state of the tidal phase. So they were all collected during the ebb tide.
Thank you. Holger Briggs asks, what is the influence of the carbon export changes on the air-sea fluxes? Should I ask Holger to clarify? Holger, do you mean?
Yeah, I mean the influence that you described was the influence on the DIC load in the river. But once it reaches the river mouth, how does it change the budgets?
So once the changed carbon export reaches the coastal regions, then how does that change the air-sea fluxes, do you think?
Oh, okay. So in the continent, like in the adjacent coastal region, so there'd be more DIC delivery. Well, during the drought, there'd be less DIC delivery to the coastal estuary, but we didn't look at the CO2 fluxes in this region. But if there's if there's lower DIC delivery, it could reduce the flux. We'd have to look into the area specifically.
I have another question. You said there's no change in the air-sea flux from during the drought period compared to no-drought period, but you did see an increase at the end, it seemed like. Can you explain that?
Yeah, so we did find in the last, I think it was the most recent 3 years or 4 years from 2018, 19, and 20, there was a gradual increase in the CO2 flux from the water to air. And this is likely because the upper estuaries, which I showed, was predominantly autotrophic in late spring and summer. But we found in those years that it was potentially shifting to dominating heterotrophic, as we found the DIC concentrations were higher in this region. So potentially the whole estuary in late spring and summer was heterotrophic, which would increase the amount of DIC produced within the entire estuary, and that could have contributed to the higher CO2 flux from the water to air. But it seemed like a non-linear response, not immediate after the drought.
Okay, thank you. I guess we have the next.
I was going to ask a question.
Oh, wait, there's another question.
So I was looking into the platform. There's still time. So, Louise, thank you for your talk. I really enjoyed it, and I think we're going to go to the next speaker now.
Okay, thank you. Yeah.
So, Eva, are you online? Ava? Okay, so the next speaker. Just getting organised now. Okay. Yeah, no worries. So, Ava, are you there?
Yes.
Yeah, okay. So Ava's going to talk to us today about burial in sediments, which is a kind of a natural extension of some of the coastal work that we've been doing. So Ava, thanks for giving this talk. We'll give you a quick warning when you're about 5 minutes out. And to everybody else in the room and online, don't forget to ask your questions in the app. And Joanna's going to pick out the most popular ones. So Ava, thank you, and it's over to you.
Thank you. So I'm Eva Ferreira. I'm a PhD student in LSR in Paris, but no, I'm not in Paris. That's why I'm online. So I will talk about the carbon sinks in product-like sediments as a troubled trigger environment. So as you probably know, the coastal ocean represents less than 7%, but it's more than about 25% of the global CO2 uptake into the ocean. And if we look most particularly at the estuaries and the river-dominated ocean margin, so it's less than 0.3%, but it represents for 40% of all organic carbon buried in the ocean sediment. So pro-delta sediments are key area for recycling organic carbon, so acting at a double trigger carbon sink. So you have a large proportion of organic matter coming from the land, from the rivers, and buried in the sediment. So you have a large mineralization in this sediment by aerobic and anaerobic processes, and that can create like DAC production. But also, if we look at the aerobic processes and aerobic mineralization, you can also produce a large amount of alkalinity. So in some cases, this production of alkalinity can make like fluxes from the sediment to the water column, and can like offset the DAC production and boost the seawater capacity to store the CO2. Yeah. So in this presentation, I will focus on the relationship between the alkalinity fluxes and the anaerobic processes using the Rhone Delta as an example. So that's the Rhone Delta in the south of France. So we can characterize this area in three big parts. So the proximal zone with a large mineralization and a large sedimentation, just like at the river mouth. The prodelta with also like a large mineralization, but lower sedimentation rates. And after the distal shape, so now more on the fifth with low mineralization and low sedimentation. We use several techniques to make in situ and ex situ measurements at the water sediment interface, but also in the pore water to make like profiles of the alkalinity and some radiocarbon and sulfate. So first, we use a new method to estimate the accumulation of sediment in the prodelta. So we use a differential bathymetry over different years, and this technique helps us to measure the accumulation at the structure scale. So in this in the RioMap project, we have made like a new cruise to collect bathymetric data in 21 and also in the 23. And we compare this to historical data from 23 and 28. So if we look at the Z meter delta, so in the river mouth, we have like the depth decreased from 25 meter to 70 to 50 over 80 years, showing like a sediment accumulation of more than 10 meters, so 55 centimeters by year approximately. So we can see that the accumulation, it's like quite variable with like 7 meters here and 5 meters here. So yeah, that's related to the importance of like the dynamic of the ecosystem with a large amount of floods, so accumulation and storm with erosion. But what we can say, it's a very large sedimentation rates, high sedimentation rates area. So now by combining organic carbon content from various cores in this prodelta, we estimate organic carbon accumulation. And what we can see is that organic carbon accumulation is a major carbon sink in deltaic sediment when we look at the different fluxes. And this highlights the need for like careful estimation of the organic carbon burial. So now if we look at the DIC, alkalinity, and oxygen fluxes from like the proximal zone to the distal shelf, we can see that we have a source of both DIC and IKT in the bottom water with a large gradient from the proximal zone to the continental shelf. We can contrast like the different zones depending on the sedimentation and mineralization rates. And yeah, you have like a lot of fluxes of JC and IKT, especially in this Z station, so the river mouth. So if we look at the same station, the station in the river mouth, the DIC and sulfate profiles in the pore water, what we can see is that we have a large sulfato reduction, so profile sulfato reduction, who represent a major like organic carbon mineralization in this area. And yeah, sulfato reduction produces like 2 mol of alkalinity and 2 mol of DIC by sulfate reduce, which correspond to the profile that we have, and which yeah can like increase the DIC and alkalinity in this area. And so now, if we look more particularly at the burial of iron sulfide, so it's a key point in the debate, because the way in which the byproducts of the sulfato reduction are removed and the oxidation stage or organic matter will change the TA and DAC ratio. So more reduced organic matter you produce, the higher TA and DAC ratio you can have. So sulfides can react with iron and form iron sulfides. And yeah, the low H2S level, along with like high alkalinity fluxes, suggests like that we have most sulfate trapped in the soil form. So we can look at the AVS measurement and confirm that FES will precipitate in these proximal and prodelta areas. And the precipitation in anoxic sediment layers creates like a strong TA gradient and TA sources, contributing significantly to alkalinity production in the sediment for water. So if we look at the biogeochemical cycling of carbon, iron, sulfide, and alkalinity close to this Rhone River mouth, so in a high sedimentation rates area. We can typically summarize the processes like this in a 1D model. And yeah, with intent FES production, creating like a sink of reduced products and a source of alkalinity from the sediment to the bottom water. So this results about the iron precipitation and the one that we have before. So we can say that we obtain like similar orders of magnitude between the alkalinity sources obtained by the precipitation of iron and the measured fluxes. So that which clearly suggests that the iron precipitation of like a Q rule in the alkalinity fluxes measured, and most particularly in this large sedimentation rates area.
4 minutes left.
And so, yeah, just to summarize, we have like ice accumulation, sediment, sediment, and this area is very active with a large burial of organic carbon and very like active organic carbon recycling zone. And this can act as the carbon sink because you have like this important burial of carbon, organic carbon, but also the way that the mineralization is done with alkalinity fluxes that can like counterbalance this DAC production and doing more alkalinity fluxes, and that the seawater can just like sequester more CO2 after that. But if we look at the DAC and alkalinity fluxes, we can see that we are more between 0.5 and 1. So we can't like really say that we have like more alkalinity, but what we can say is that they can limit the acidification potential of the released DAC without creating an additional sink for atmospheric CO2. So that's all. Thanks.
Thank you, Eva. We have a question online from Richard Sanders. How secure are these sedimentary sea stores, carbon stores? Will they eventually get covered by vegetation and get stabilized, or do storms or dredging periodically erode them?
So you have, I don't really know like the rates of sediment taking by the storm, but you have like a very large sedimentation rate. So even if you have like storms, it's not like big storms, and they don't take like too much like sediments. So yeah.
A follow-up, hold on.
Thanks, Eva. I thought these data are just really amazing. So if you look at a map over time, presumably the delta is actually just getting bigger, geographically.
Can you repeat just the beginning of the...
Is the area of the delta getting larger?
No.
So these really high sedimentation rates, eventually, does it just get incorporated into the land?
Oh yeah, but it's more like in the like vertically. that you can say? It's like vertical accumulation on the delta, like more depths, or less.
And Eva, Richard also asked about dredging. Do you have any idea about dredging activities in the area? How would they disturb that?
Yeah, I know that they have dredges, but I don't have like a real like study about the real like impacts and the real place. So we don't really know if dredging will just happen in the area that we study or not, but probably, yeah, I don't really know.
Okay. Oh, we have another question. Thank you very much. Carol Kulinski asked, have you tried to quantify denitrification as alkalinity source?
It's in, I will try. I don't really like, I don't have like the result here, but yeah, I think it's on the project.
Okay, thank you very much.
Thank you.
We have one more speaker online. Maybe Richard will introduce her.
Okay, so Michelle, are you there?
Yes, I'm here.
Okay, great. So I'm really looking forward to your talk about the Gulf of Trieste. So once again, everybody, Joanna will give a 5-minute warning, and you can ask your questions in the chat. So are you ready? We good to go? Over to you. We haven't got your slides up yet. We're just loading the slides now. Are you still there? Is he still there? They're doing it.
Can you hear me?
Yes, we can hear you.
Yes. On the karstic.
We don't see your slides yet, though.
Sorry, sei in condivisione. I'm interrompi. I do again because okay. Yes.
So that's perfect now.
Okay. Okay. So I go to the first. Sorry. Why don't?
Yes, you start whenever you're ready.
Yeah, but I cannot move the forth and back to the slides. This is a problem. Why is not going? Sorry. Yeah. Okay. Now it's going.
Oh, brilliant.
Okay. Yes. So I will talk about the influence of karstic rivers on the South and Adriatic waters and on the CO2 system. So we collected the data on 3 sites in the Gulf of Trieste. yes, on the Gulf of Trieste. Can you hear me?
Yes.
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 Yeah. And in these three sites is a big delay.
We seem to have a delay, I guess.
There is a big delay in the. Okay. So we collected samples. No, is a lot of delay. Yeah, but there is a big delay.
How about you just continue talking and don't pay attention to anything else? And I will not, because you're the last speaker, I will not say anything about the minutes. So just go ahead through the talk.
Okay. The objective.
Or maybe you can remove your screen sharing and share again, please, Mr. Gianni. Put it in diaporama mode. Yeah, it's moving now. Otherwise, we can share your presentation here locally.
Yeah, maybe it's better.
Okay. You just have to tell us when to move the slides, okay? Okay. You can start talking and let us know when you want us to move the slides.
So the objective of this research was on the carbonate system.
Problem might be that he's using his earphone. I think the delay might be in his voice.
There was a suggestion from the audience to maybe take your headphones off. Does that help?
Mr. Gianni, do you hear us?
Yes, I can hear you, but yes, I hope.
We hear you. We hear you well. Do you see your slides on my camera?
One moment, I don't see.
My camera.
On your camera? No.
Yes, you see your slides.
Yeah.
Okay, so fix on that slides, start talking, and let us know when you want us to move the slides, okay? You can go forward. Go.
Okay, move
Go
on
ahead.
the second, on the, move on the next slide, please. Yes, so the northern Adriatic and the Trieste Gulf is the northern part of the whole Mediterranean Sea, and is an area where the warming is between the highest of the Mediterranean, as you can see in the sea surface temperature of the Mediterranean, and the warming is higher than 0.4 degrees per decade, and is also a quite productive area, as you can see on the left with the chlorophyll. It's a shallow area with a maximum depth of 25 meters, and is influenced by riverine inputs, mainly from two rivers, Isonzo and Timavo, and which watersheds are karstic, especially for Timavo River, and we have a lot of influence of due to wind, especially northeasterly winds that are strong in mixing and also in determining the formation of the dense water, which are the densest, among the densest water formed in the Mediterranean Sea, and they flow southward till the deep, till the Ionian Sea, and then they contributed to the deep waters of the Eastern Mediterranean Sea. And we have a very strong seasonality and with strong temperature range from less than 8 degrees to more than 29, and salinity ranging from less than 20 to about 38.8. So we have a strong stratification during spring, summer, and these are the main features of the Gulf. So you can pass to the next slide, please. So we have a lot of discharge from northern Adriatic rivers, as you can see in this slide, where the input of total alkalinity, DIC, and bicarbonates were estimated for most of the northern Adriatic rivers. And on the northeastern part, you can see the Trieste Gulf with the two rivers that were under investigation in the study period of this research that is still ongoing. So there is this relevancy input of alkalinity is in the Northern Adriatic is estimated to be about 10% of the whole alkalinity coming from rivers and from the Black Sea into the Mediterranean Sea. Go to the next, please. So the sampling were carried out monthly during 1 year and a half, and as you can see in the red are the samplings, but in this period there was the average flow of the rivers was low. In fact, we had a freshwater discharge that was about 50% of the average of previous years, and also the estimate that you can see for the 2022 was in terms of alkalinity and total CO2 was about 50% of the mean discharges for this during 2007-2014. Go to the next slide, please. So in the Gulf of Trieste, as also the Northern Adriatic, is influenced by the incoming of southern water that are coming from the Ionian Sea. And these waters are changing their characteristics depending on the Northern Ionian Gyre. So in the Northern Ionian Gyre, we can have an anticyclonic circulation during which the inflow is mainly of Atlantic waters and in the surface. So waters coming through Gibraltar from Atlantic. And when it's reversed to a cyclonic circulation, the water are coming from the Levantine Basin, which is a yellow arrow on the right part. So during the samplings, we were in a cyclonic phase, as you can see from the graph below, which shows the monthly vorticity of the Northern Ionian Gyre. And this was meaning that during these phases, we have waters with higher salinity, which are flowing into the Adriatic Sea. Please go to the next slide. So in the Northern Adriatic and the Gulf of Trieste, we have a negative relationship of total alkalinity with salinity. due to the elevated inputs of bicarbonates from the watershed through limestone and karstic areas. Also, if we were in a period of a very low freshwater discharge, anyway, the relationship was significant. And in the next slide, I will present the difference between total alkalinity and total CO2, which can be used as a proxy for carbonates, pH and omega aragonite. But also as a proxy of the rebel factor, which is, let's say, the buffer factor. So I will principally talk about this to show the buffering capacity of the system. Please go to the next slide. So in here you can see the trends on two of the stations. So one very coastal and another Paloma in the center of the Gulf of Trieste. And for the surface waters, we have an increasing salinity trend, and also for the bottom waters. These trends are estimated from 2009 to 2022 for Paloma Station, and from 2011 to 2022 for the C1 Miramar Station. And if you see the graph below, you can see the trend in alkalinity, which is significant mainly in the bottom and waters, and this is related, we suppose, is related to the ingression of this southern water of Levantine origin. Go to the next slide, please. So here are the time trends, both for salinity and alkalinity I showed in the first slide, but also for TCO2, and also for the difference between alkalinity and TCO2. So the TCO2 is increasing in bottom waters, especially in the shallow site, Miramare, more than in the offshore site, where the apparent oxygen utilization is decreasing. So the minus TCO2 decreasing trend in the shallow site and increasing trend in the offshore site could be due to a different behavior, due to the reduced degradation of organic matter in the offshore station, which is more influenced by the main rivers, that is the Isonzo River, which in this period had, in the recent period, had reduced discharge. Go to the next slide, please. Here you can see the variation with depth of salinity in the upper graph and alkalinity in the graph below during the study period, to the 18-month period from 2021 to 2022. And you can see the lowering of salinity is corresponding to a higher salinity in surface water, and at both sides. Go to the next slide, please. Here is evidenced the difference between alkalinity and total CO2, and where it's possible to see that we have the highest values of this difference. So the highest values of buffering capacity is in correspondence of the surface water influenced by rivers. Whereas in the bottom waters, where we have the lowest apparent oxygen, I mean the highest apparent oxygen utilization, we have lower values of this buffering capacity, as well as during the winter dense water formation, so during winter cooling, because during winter cooling we have an absorption of CO2 from the atmosphere, which is increasing the total CO2. So, and on the right, you can see the difference of different water masses detected, where you can see that the buffering capacity is higher for the waters under river influence with respect to the southern waters, and with respect to the dense waters that are the lowest in the graph. Go to the next slide, please. So in this slide is presented the relationship between the C13, delta C13 in DIC and the apparent oxygen utilization, where there is with a decrease of apparent oxygen utilization, I mean with a negative value, which is meaning increase in primary productivity, we have an enrichment of DIC, which is due to the preferential uptake during photosynthesis of the lighter isotopes. Whereas the contrary is occurring with increasing respiration in bottom water, which have the highest apparent oxygen consumption. Go to the next slide, please. In this slide is underpinned the effect of upwelling of CO2 rich bottom waters during summer at the coastal site of Miramare, where you can see that there is in August in the black rectangle there is an homogenization of the water column and a strong increase of the PCO2 and a decrease of the difference between alkalinity and total CO2, so a decrease of the buffering capacity. In the graph below to the right is possible to see the temporal variation of PCO2 in microatmosphere, is not milliatmosphere, but microatmosphere, where is evident the seasonal variation at the bottom with an increase of PCO2 during the stratified period, and also some events of this upwelling of waters due to this boreal event, which are here underpinned by this graph of wind direction and wind intensity. That was a changing of the direction during boreal wind blowing, just when we were sampling, and this explained the upwelling of bottom waters. You can go to the next slide, please.
Michele, how many more slides do you have?
Yeah, I'm at the conclusion. So to conclude, a high variability in the discharge affects the delivery of carbonates and alkalinity associated to the fresh water, which contribute to buffer the waters in the Trieste Gulf, but also in the North Adriatic is happening the same. The decrease of freshwater discharge and the increase of activity in bottom water indicates that the increase in total alkalinity observed was not of river in origin. During the study period, the low river discharge in the cyclonic Ionian gyre favored the ingression of high salinity southern water characterized by low total alkalinity minus total CO2, so by a lower buffering capacity. This might reduce the buffer capacity if these two things are coupled, but still further investigation are ongoing. Both wintry dense water formation and events of coastal upwelling of bottom waters rich in PCO2 during summer contribute to lower the buffer capacity. Further ongoing analysis of samples collected during a period of relevant discharges in spring of this year will allow to better assess the influence of freshwater discharges on buffering capacity in the Trieste Gulf. The next slide, and these are so here is also a poster in the poster session about the influence of the rivers in the Gulf of Trieste. Next slide is the acknowledgement. So thank you for your attention, and I'm displeased for the problems with the delay in the speaking.
So we do have one question in the chat. We're out of time, so we'd appreciate the quick answer. Are your findings only site related with the particular circulation pattern, or can they be explained to a larger area? Expanded, sorry.
I mean, the effect of the changes in circulation in the Ionian Sea affect different areas in different parts of the Mediterranean, and also in the Southern Adriatic, and recently was also seen more clearly also in the Northern Adriatic Sea. And now we are investigating because this has, let's say, some changes that can be also decadal or a little bit less than decadal, these changes of circulation in the Ionian Sea.
Thank you. And Richard can conclude.
Well, I'd like to thank all the speakers. Michel, thank you for your talk. We're going to go for coffee now. So hopefully you'll be able to join the rest of the conference or another bit of the conference later this week. I'd like to thank all the speakers. I'd like to thank my co-conveners, Holger, Carolina and Joanna, and thank you for manning the chat. We're back in another session in a little under half an hour. So, and also I'd like to thank the technical guys at the back for fixing a few issues along the way and keeping us to time.
Can I get your attention, please? So welcome to this session here today on greenhouse gas fluxes at high latitudes and climate human-induced feedbacks. And this is organized on behalf of the EU project called Green Feedback. And today, and Sophia and I will be chairing this session. We're also going to be having session tomorrow as well. We're going to be having 5 presentations today. And I want to just remind you that the questions that you have, you should put it online into the chat so that we can ask some good questions here for the presenters today. And we're going to be starting with Sophie. Yeah? So if you're welcome to come up.
Oh, there's no screen, I cannot see from here. Okay.
Yeah, and you know, you press and do. Yeah.
Good afternoon, everyone. Xue Li from University of Helsinki, university researcher. Today I will talk about the temporal and vertical variation of methane turnover processes from northern peatland using stable isotope evidences. First, some brief background. I guess you are very familiar with it. Methane is a potent greenhouse gas. It's the most potent greenhouse gas after CO2. It accounts for about 1/3 of the climate change effect. And in the recent years, there is like a dramatic increase in the atmospheric methane concentration. So you can see here. Oh, sorry. Either something. No, yeah, something here. So on the left-hand side, the y-axis is the methane concentration in the atmosphere, and the x-axis is the years. So in the 800,000 years, in the past, the methane concentration historically is about between 200 to 800 ppb. So it's like showing the historical, like the natural background of methane concentration in the atmosphere. Well, from the onset of the Cultural Revolution, that you can see from the right-hand side of the figure, there's a like rocket increase of the methane concentration. So we want to know where those methane in the atmosphere come from. So there's a very nice review of from this global methane budget that we can see from the red arrows shows anthropogenic methane emission sectors, which I will actually have another talk tomorrow, just a little advertisement tomorrow. I talk about that in this session 18, tomorrow afternoon. But today I focus on the natural source of methane emission, which wetland is the biggest emitter as a natural source. And from here we can see there's, well, it should be an animation, but it's a PDF, so you don't see, but you can imagine there is a lot of changes, seasonal variations of the methane concentration, and it's moving from the northern hemisphere to the southern hemisphere, and it's changing related to the temperature change. So it's a lot of uncertainties. We need to understand what, how methane emission change over the season, so that we can predict better how it's correlate with environmental variables, and so that we can put them into the ecosystem models, so we can predict better with the future climate scenario. Since we are talking today, our session is about the northern latitude, so I mentioned that peatland is dominant wetland type in the northern high latitude. Just briefly about what is a peatland. So it's type of wetland, as you can see, is very high water table, so it's a slow decomposition of the organic matter. You can see here it is on the bar. It's like you can use it as a historic record to study in the past what the climate look like. As this variation between the Carax and the sphagnum matter, and yeah, it's very slow decomposition. So you can see there's human remain is from 2,000 years ago. It died somewhere in somewhere called the Lindau Moss near Manchester, and it's preserved very well. Now you can see it in the British Museum still. So there are some specific features of peatland. so the plants develop parenchyma, which is kind of tubes inside the plants to transport oxygen to the root because the root need oxygen to respiration. And there's also sphagnognosis, so it's kind of a spongy layer that is, it could absorb a lot of water. So comparing, I think about 20 times or 30 times of its dry weight, it could collect the water. So that, in that sense, the peatland is a bit resistant to the drying because it can regulate itself, even if it's a bit drier or a bit more wetter. Just some brief background. So talk about the seasonal variation. So there's a lot of studies about the seasonal variations of methane emission using eddy currents towers or chamber measurement. But the emission is net outcome of the production and oxidation, basically. So if you want to really understand how it varies seasonally, we need to also to go decompose it into different processes, not just see the outcome. And so mostly in the past, when you study the processes, production, oxidation potentials, people use incubation a lot. So you take the peat from the peatland and then go to incubate in maybe 15 or 20 degree, and you see how the microbes, the archaea, can produce or oxidize methane. But you separate them at the time you take them away from the field. Basically, you just separate them from its original status. So there's some disturbance. And stable isotope actually provide a very useful tool that help us to study these processes directly in the field. So you can see here, there's a nice summary of all the methane processes related certain C fractionation, which is, yeah, so maybe I don't go to very detail. So that's isotope signature is the ratio between certain C and 12C, and we use it to show how much certain C, how much 12C there. And in each process, there is a bit fractionation. So either some 12C is consumed or goes faster or consumed better. So there's a different delta C value in the methane along the processes. And by understanding that, by looking at certain C value at a certain time, like we're taking a snapshot for this current status of the processes. And if we can measure it continuously over a long time, frequently, so basically we can understand better the seasonal variation of different processes without intruding the ecosystem living things there. So that's our study scheme that we want to study using isotope, and we want to study with high frequency. So that's the first setup ever made for this kind of scheme. So we have below ground tubes to measure methane, CO2, and their isotope. And also above ground, we also measure the certain C from the emitted methane. So like we have a continuum from the soil to the atmosphere. And also we also measure using isotope labeling technique to actually to target, because there are some, in the natural abundance approach, it's nice, but there are some processes it's co-occurring and it's moving the isotope value in the same direction. So we want to separate those processes more specifically. So we did also the isotope labeling, but it's not a part of this presentation. But it's just to tell you how we can combine this isotope method together to complement each other. So let's see a study site. It's C. caneva. It's Icos type 2 research site. So it's it's very homogeneous comparing to the box at the fen site. On the other side, we have the bog, but this fen site is relatively homogeneous. There's a carex, aleuforum, and there's a sphagnognosis as the carpet. So snow cover is from November to March, and there's spring zone, and there's a spring snow free time is from June to October. The average peak depth is from 2 to 4 meters, I think, where we measure where we have these tubes, which you can see. Is the red cross is here. So that you can see.
The online people cannot hear you. Yeah.
Okay,
Speak
sorry.
in the mic, sorry.
So the black cross is the eddy current site, eddy current tower. We have it there, and from there the blue dashed line indicate where the tubes are. So it's a 50-meter long tube. There are 3 of them, as you can see here. It covers quite a big area. So the idea is we can average the different microtopography. So we have the, sorry, oh, okay, it's very. Then let's go to the. We ask how methane production pathway vary temporally and vertically, and we ask what's the driver of the seasonal cycles we have seen. So there are some hypotheses. If it's only driving by peak temperature, we should have a scheme like that between the pore water concentration and the isotope of the pore water. If it's driven solely by temperature and substrate availability, then we should see increase like that. But if it's driven by both temperature and substrate availability, but there's a time lag between these two drivers, we should see this kind of time lagged hysteresis-like behavior between these two variables. And in the end, we want to ask what the methane transport pathways varying temporally. Okay, briefly, some result here. It shows the environmental variables, the PAR. There you can see the seasonal variation. So we measured from the March 2022 until March 2023, a full annual cycle. And the soil temperature shows it reached the highest peak a bit earlier in June, and then the blue dot shows the LAI of the Carex species. So the plant produced the maximum acetate input for methanogenesis a bit later, reaching a bit later. So that we can see later in the result. And here is the concentration in the soil pit profile. We can see in general it follows that winter is lower, summer is higher. However, there's also change of the vertical profile between the winter and summer. And in the winter time, the 50 centimeters, so the deepest tube has the highest, and then followed by the 30, and then followed by the 10 centimeter. Well, in the summertime, the 30 centimeter tube has the highest concentration, while 50 is a bit lower, which is understandable because 30 centimeter is about the where is it the rooting zone, where the root produces most acetate as a substrate for methane. While the low concentration in the surface can also be because it's sometimes water table in the summer goes below below this 10 centimeter tube, so that the diffusion to the air is very fast, so we have very low concentration. Then we can see the isotope value, which is also follow the concentration that is highest in the summer and the lowest in the winter. However, there is a flipped vertical profile can be observed between the winter and summer. For example, for here in April 2022, we can see the lowest tube has the highest isotope signature, while the highest is the lowest. But in the summertime, it's opposite. So that shows there are some, we're still trying to understand why it's like that. So in the summertime, it's easier to understand because acetoclastic methanogenesis, which is using the acetate as a substrate, it tends to result in the delta tendency a bit higher. So we can see that's in the surface, it's more towards the acetoclastic methanogenesis. But while in the winter time, why it's like that? There are several hypotheses we can think of. One is because of diffusion, because it's highest concentration in the lower tube, while the methane diffuse up. So when it's diffusing up, it tends to get lighter, the isotope value. Another Another explanation is it's more towards heterotrophic methanogenesis, and it's also temperature specific. So because in lower soil is a bit warmer in the winter, so it has a bit effect on the signature. So we are still trying to figure out what's maybe it's a combination of several different processes going on. That's, if you have a better idea to explain, I'm really happy to discuss with you. So, well, maybe that's how we calculate the surface isotope value. Maybe I just skip it. So you can see the result first. In the pore water, so if you remember the second question we asked, what's the driver of seasonal variation of the processes? And we can see clearly there is this hysteresis-like behavior as the third hypothesis we have. So it showed there is a time lag response to methane, to soil temperature and the trophic level, how much acetate there is. And sorry about that. So there's a lot of figure already that I don't have more. Here you can see along the line, it shows the evolution, it basically shows the methane transport pathway. Along the line, the evolution, and here is plant transport because it's lighter, isotope goes faster, so it's going, so it's towards the lower half of the figure, and here is the diffusion because through diffusion there's oxidation, and oxidation normally enrich the residual methane isotope. So just show you that from here we could understand a bit through which pathway it is transported. So here are takeaways, and thanks for everyone who has contributed to the work, and thanks for your listening. Happy to discuss if there's time.
No, thank you so much for your presentation. We went a little bit over time, so we have to go to the next presentation right away.
Yeah.
So Elodie is going to be talking about wintertime methane fluxes in boreal and Arctic peatlands. 15.
Hi everyone. So we keep going on peatlands in boreal and Arctic region. And thank you, Chu-Hei, for the nice introduction on peatlands, so I don't have to go much more into detail about that and on methane processes, but we're looking at methane flux during the winter time. And that works is a team effort from people from different institutions that we all working on under the project Green Feedback. And I want to start with the definition. So what do I mean by wintertime? There is multiple use of term in the literature. About wintertime, is it wintertime, cold season, or non-growing season? Everybody has a definition, and Ito and Al has provided a summary of 3 definitions. That the first one is sub-zero monthly mean air temperature. Some other studies are using non-summer months from September to May. And another definition is mid-winter months from December to February, when the soil temperature is below 0. So for that talk and that preliminary work, I choose maybe not the best definition, but the easiest to work with the data set, which is having a fixed period from September to May. And here are a few literature work that have been done on site on the 3 different continents, showing that methane emission from wintertime contributed for how on the same amount or nearly the same amount during winter and the growing season, the summertime, in high latitude regions. So I'm not going into detail of each of those diagrams, but each of those diagrams, that's what they show. And there is that other study by Ito et al., a recent study, that look at the wintertime emission by global methane budget model, the process model, and they compare the output from the process model with the upscale edicovariance product from Peltola and al. And their main conclusion is that ecosystem models are underestimated cold season emission. So what we are interested in for that study is to improve ecosystem model and their ability to predict future Arctic and boreal methane budget. And for that, we selected two ecosystem model, Orchidea and LPG gas, and as a preliminary work, we pick 7 sites that has data, so they don't have continuous data, but they have some data over the period 2005 to 2023. And those two ecosystem models are representing as well as the production of the methane, the oxidation of the methane, and the 3 types of methane transport. And so what I'm going to show you is the result of the model in comparison with the site observation to verify the capability of the current version of those two ecosystem models, and trying to see if model that can split by their setup the different transport process. So what is out of the scope of that presentation is a spring burst. I didn't look into detail about how the spring burst was modeled. And I divided the year into 3 periods. The summer, that is from April to August. The winter, so from September to May, with 0 cotton from September to December, which is of the fall period, where the soil temperature is hold around 0, and the freezing period from January to May. So those are the preliminary results. As you can see, I didn't have the time to process all the site data, so there is only 3 out of the 6 sites. And the first thing that you can observe is that for Copperfield and Zackenberg, there is no 0 cotton methane emission data. And it doesn't mean that there is no emission on the site. It just means that colleagues couldn't go on site because of the snow cover. So there is a lack of data because of the accessibility to the site. And on the 2 models, you can see that for some sites, the proportions are quite similar, and for some other sites, the behavior is different. And that is due because of the formulation of the model, and also because we couldn't use the same forcing. I ran into a trouble with Orchidea, and I couldn't run the same forcing than my colleagues. And another thing that I want to point is for Zackenberg here, there is no pie diagram. That's a diagram of the snow depth because the model simulates too much snow thickness. And so because the thickness was 2 meters all year long, so they didn't have methane emission for that site. So another process that is observed on site, it's the soil temperature, the correlation between soil temperature and methane emission. And during the warmer season, the temperature increase and you have an increase in methane emission, and then you have that fall period with zero cotton time where the methane is decreasing, and then you have a minimum. And depending on the year and on the site, you have different behavior, different correlation, and that other study also show the same, a different pattern. So here are the preliminary results that we have for the 3 sites and of the model. So there is some work here to do to understand if the correlation is good or not, because it's variated a lot for every year. And also we have to harmonize the data, because as you can see here, for example, for Sikaneva, there is some methane emission data, but we are lacking the soil data. Okay. Let's keep going. Then I look a little bit into some detail about the zero cotton season in the fall, because there are some processes that have been hypothesized during that period, where you have the surface soil temperature that is decreasing, and so the surface that is freezing and accumulated or concentrating the methane into slightly deeper layers. And so to look at that, I plot on the same diagram. So the color plot is temperature, the soil temperature profile, and then in yellow it's the zero isotherm. In red is the maximum methanogenesis production, and in blue it's the methanotrophy, the maximum of methanotrophy. Okay. It's easier to look at an average over the period over 1 year. And what we can see here, depending on the site, the isotherm is going deeper in the summer or in the winter. And for example, here on Hulanka, you have, so that's simulation result. You have methanogenesis that is also deepening and with methanotrophy. Then in LPG gas, the formulation is slightly different, and the variation of the depth, it's less sensitive. So what we can see also here is that that variation is dependent on the snow cover. And the last processes that I looked at is a distribution of the different gas transport between diffusion in blue, ebullition in purple, and plant-mediated transport in green for those 3 periods. And so you can see that for some sites, we see different proportion. So for LPG gas, it makes some sense. And for orchidea, there are some sites where you don't see any diffusion. So it doesn't mean that there is no diffusion, it's just because the plant transport and the ebullition is stronger. And those 2 distributions are connected to the way that we formulate the processes, depending on whether or not the gas is going through the snow, or if the gas are diffused into the snow. Okay. Another thing that I want to point out is here you have the snow thickness and the 3 different periods that I defined. And depending on, for example, for that site, you can see that the snow cover is not during the whole freezing period. So you have the April and May where you have a plant transport that is emitting, that you can see in the pie diagram of the previous slide. And so that's not the best way to look at the data by defining some fixed period, because the snow cover for each site is going to be different. To conclude, so we look at 7 sites so far, and we need to work on the harmonization of the data for the winter time. The ecosystem model that have been used, so the model Orchidea and LPG gas, are underestimated at global scale. That's the work from Icos that I showed you at the beginning of the presentation for wintertime emission. And what we have seen through that preliminary work is that those processes the emission are consistent with the sensitivity to the snow cover influence, because the snow cover is going to influence the soil temperature, soil moisture, and the different gas transport. And I just want to acknowledge the funding agencies, so the European Union for funding the Green Feedback Project, the different institutions, the Icos and the other monitoring site, and the high performance computing facilities. And thank you for being here.
Thank you so much. I just want to remind you to add questions to the chat. I know it's late. I know you just ate some cake downstairs, but you had coffee. So I hope that it evens out. But remember, please.
Yeah. There's one question here for you, Elodie. Thank you very much. The question is from Tanya Liebman, and she asks, it's generally thought methanogenesis is more sensitive to temperature change, but if I understand correctly, methanotrophy looked like it had more seasonal variability. What do you think about that? Could it be partly responsible for low emissions?
It's difficult to respond with a short response, because everything is interconnected. So yes, all the study on site or in the model have shown that methane emissions are mostly sensitive to temperature. But we also take into account the water table, and that has an effect. And so that's what is adding to the effect of temperature. And so for methanogenesis, it's a balance between that. So in those very high latitude region, it's even harder because the water is not always liquid. And well, temperature and water has to balance themselves in different stage. So that's why it's not easy to respond clearly to the question.
Great, thanks, Elodie. I have a question for you. In the beginning, you talked about cold season definitions, and I was curious, this type of definition, how will that affect your analysis? So you showed in one of the last slides, for example, the snow cover that varied from site to site. Now, is there any way you would choose a different way of defining your cold season?
Yeah, I saw some paper where they start the cold season. Well, no, they compare the methane emission, taking as a starting point the melting of the snow, and then you can compare the behavior, the trend of methane emission, and I think that's the best way to do it. I don't know how to do it for wintertime. Should we take, the beginning of minimum thickness, maybe? I don't know. We will probably think about that with my co-author.
Great, thanks a lot.
Thank you.
Thank you. And then I would like to invite Margaret to the stage. She's going to be talking about high emission of CO2 and CH4 due to active layer warming in Arctic tundra.
Thank you. All right. Do I start it?
I think they started behind. What? Don't.
Okay, tell me how I advance the slides and point. Can I use this?
Yes.
Okay, sorry, you can. All right, thank you. Yeah, I wanted to share results from a manipulative experiment in which we see that warming the whole soil active layer in a permafrost Arctic tundra results in very large CO2 and methane fluxes. So as everyone knows, we're interested in this question because if warming leads to more greenhouse gas emissions, then that's a really powerful contribution to a positive feedback to warming. But the specific goals that we wanted to look at was, could we quantify the direct effect of warming in situ on these greenhouse gas emissions? And to do that, we wanted to also develop and then test a sort of novel warming approach to let us manipulate the whole soil profile, which is shallow because it's permafrost, in situ without having to do a manipulation and the sort of artifacts that we heard about from that. We worked in Barrow, now called Utqiagvik, Alaska, which is at 71 degrees north. And we worked, this is a polygonal tundra environment, and this study was conducted in the high centers of polygons, so it's a bit well-drained. What did we do then for this micro-warming, I'll call it? Oh, I'm like, one moment, please. Well, okay, I'll try not to wave around too much. So I'm calling this a micro-warming because what we did is we have a little PVC mesocosm, and inside each one we put a single metal rod that had a resistance cable in it. And we could heat the soil then down into the permafrost. The permafrost depth was about 45 centimeters here. We got a pretty constant warming. And this was all controlled by solar panels. We had 223-watt panels that managed to work through the growing season and just into the fall. Okay, let's see, okay. And so we had 4 blocks, each one with a heated and a control pair, kind of shown here in an aerial figure. Actually, this is a little distracting. How do I turn that off? Maybe I don't. Ah, great. Yeah. Thank you for your help back there. And so we had 4 blocks, which I was just showing you. Do I need to advance that? But I don't know how to go to screen share again. So I'll let you do that. So I was going to start, I'll just add, while we're figuring that out, that then we measured CO2 fluxes. I'm putting this down. I'm not doing this. That's not me moving that. We measured CO2 fluxes with opaque chambers connected to a Los Gatos research instrument that is somehow called ultra-portable greenhouse gas analyzer. But it's not. But it worked well. It worked well. And then we also had an opaque chamber. Awesome. Thank you. I will try not to cause more problems here. And I'm not going to talk about NEE, but we also had a clear chamber. The Los Gatos research measured methane, also, which I may have forgotten to say, and sort of doing that. Then we also conducted a shoulder season experiment. I'm not sure where this fits, Elodie, in your gradient, but it's after snow and ice had accumulated on the surface and made kind of an impermeable layer. But what's important to know about the shoulder season here is that the subsurface is not frozen yet. There's a thawed layer between that snow and ice and the permafrost. It's called a talik, and microbes are still active in that zone. You may not see greenhouse gas emissions or gas emissions, but they are active there. So first, I just want to show you a few results on the temperature that I think it worked pretty well. If you start, this is orange is heated, blue is control, and the gray is the difference in temperature, and we'll use the same orange and blue for treatments throughout. Starting at the 5 centimeter temperature at the top, you can see that this warming was able to warm but allowed for natural diurnal cycle variability. And if it rained, you got that. So I think it was a bit of an improvement over a laboratory incubation, let's say. I don't know that it's perfect. And then if you look deeper, what you can see is that we thawed above freezing earlier in the year, just like you'd expect, and even down into the permafrost, this system was able to thaw the permafrost, which you see at 50 centimeters. And then if we look across the whole profile, just at the average over the 2 growing seasons that I'm talking about, the average warming was 3.8 degrees. We had set the target at 4, but you could just adjust that if you really wanted to get right at 4. It was not possible to evenly get warming of that amount at 5 centimeters because of the exchange with the atmosphere of energy and a little bit of rainfall. And then at 50 centimeters, we didn't have quite that much warming because you have a phase, energy going into phase change and advection of thawed water. So now I want to show results on CO2, and then I'll show methane. So for CO2, this is showing CO2 flux, and the bars are the average fluxes across the plots on each day of measurement. And ignore the upper right just for a minute. These 2 left-hand ones are showing that on every day that we had warming, we saw an increase in CO2 flux. Not surprising, it was about 30%, which is actually fairly high temperature sensitivity. And then, let's see, we can look at the shoulder season. So now we're getting about 10 times as much flux when we thawed. And this picture is showing, it's really hard to see, but inside is a little heater cable coming out of the soil. It's about a 10 centimeter gap that it caused in the snow and ice. So I think it just let a channel form so that CO2 formed in the talc could come out. I don't think, in the annual budget, I think this would have maybe come out in the spring. So if you were measuring year-round, you would have seen this. But it was just a nice controlled way to say, yep, warming, or this could have been a rain on snow event. Anything that causes a rapid thaw would give you a pulse. So we could estimate a Q10, or a measure of temperature sensitivity, from these data, and we can do it in 2 ways. The colors in the figure on the right are showing different months. So you can see as different months went on, it got warmer, not surprising. The circles are heated and the triangles control. So a lot of people have done this, and we can do it, fit a curve to these data, and that's the Q10 fit in the upper left there, or the equation for it. But we know that other things also co-vary over the season. So as it's getting warmer over the season, you're also seeing more plant growth. We also have more thaw depth. So there's more carbon available in general. The other way to do it is to take that heated and control and compare on each day when other conditions were exactly the same, and look at what was the effect of temperature. So that's the lower one. So here's the 2 numbers that we got using all the control, just the control plots when the heaters were on. Very high Q10 of just about 7 for that ambient or seasonal variation. And it's just saying, I think it's saying that that covariance of plant growth and more thaw depth really amplifies that temperature response that you would infer if you didn't think about it. I think we know that intuitively, but this is like a nice way to compare in a controlled setting. Instead, if you use that controlled treatment difference, you see a Q10 of 2.8. And just, I should say, if you don't use this horrible measure of Q10, just to say that it's a measure of how much a rate changes with a 10-degree warming. So a Q10 of 2 would be a doubling for 10 degrees warming. And 2.8 is still quite high. So I just want to show you what that might mean. This is using CESM. It's a US developed community model, Earth system model that's used in CMIP analyses. We just used one for this. So on the left is showing the soil temperature projected at different years for different scenarios of emissions. And then on the right, I'm showing the change in Arctic ecosystem, and that's Arctic temperature. And then we get change in Arctic ecosystem respiration for either Q10 of 2 or Q10 of 2.8. Q10 of 2 is solid line. We started with what the model projected for 2015 fluxes, and then we just incremented this. So think of this as a back of the envelope illustration of how important the difference in Q10 is. Starting in 2015, we incremented each year with the temperature on the left and then either the Q10 of 2 or 2.8. So I think you can see the difference between the dotted line, 2.8, and the solid is quite large, especially in the higher emissions scenario. So that's the difference that would be implied. And we use the Q10 of 2 because that's the sort of emergent Q10 you get from this model over time. So now I'm going to turn to methane. Here we have a kind of different story, but I think also really neat, really interesting. This area was a very low-level source of methane because remember we're on this high-centered polygon, not in the wet trough areas that have aranchromatosis plants and so on. But the warming, perhaps because it dried the soil a little bit, reduced the low methane emissions to really zero. We can detect that that level of methane uptake is not just zero, the little negative orange bars. So that was true in both years of measurement. But then if we look at the shoulder season experiment, the methane emissions were 200 times that growing season number. So again, it's saying, I think it's the same thing. In the thallic, the microbes were active, producing a lot of methane, didn't have an easy gas diffusion pathway, but once we warmed, then it did. But I think an important thing about these kind of escapes is that probably if this had been a slow, gradual warming, microbes would have been active, and slow diffusion, you get a lot more oxidation. So I think this kind of escape represents a net emission of methane to the atmosphere. CO2, I think it would just stay in the thallic and go out. Like, just makes it hard for us to measure. This is actually meaning that with these pulses, I think we get more methane emission. And wanted to say what we believe is happening. Maybe this is a little bit of a busy slide, but on the right is what I was just talking to you about, the really high emissions with warming. So what we believe is happening is not that microbes are suddenly turning on, and pumping methane, but that it is building up in the thallic. I guess I've already kind of hinted that that's what I think is happening. We have some other evidence as well. So the upper left is from our eddy covariance tower, also in Utqiagvik. And that I'm showing here, and I apologize because it lost the x-axis somehow in the translation, maybe to the PDF. But this is a spring pulse. That pulse is a 2-week period when we had a rain on snow event, really rapid melt and thaw, and we got a lot of CO2 and methane. The methane that came out here added 6% to the growing season budget of methane emissions that we measured across the whole site. And this included troughs, so this was a wetter, higher methane. It was really a substantial amount. It's like a winter, and fall activity finally coming free. And then the lower left, we did a controlled laboratory core thaw, where we sort of slowly thawed a core, and again, that we had collected in the winter. So it had already built up gas in the thallic. And you see a lot of release. As far as I know, this experiment on the left or on your right is the first one to sort of, in a controlled way, demonstrate that this is what's happening, that it's built up and that you can turn it on just by thawing. But I think it'd be really easy to go out and do a rain on snow experiment. You could get a sled to carry the water just to make it easy, but you could do it, and then we would really know. Because then you could wait a week, do it again, to get just as much or less. Like, how much is this? We didn't continue. We didn't know what was happening when we were doing the measurements. Yeah. So then in conclusion, first thing is I think that the heating approach worked pretty well in an area that's pretty rugged and doesn't have line power. So this was great. We could do this. And the warming increased growing season CO2 respiration by about 30%, and we estimate that's a Q10 of 2.8, which is pretty high. And I want to say this is an immediate direct effect, right? There's no change in vegetation that will happen with warming, depletion of substrates, major changes in geomorphology that will happen in the Arctic with prolonged warming. This is kind of an immediate physiological effect of the microbes and plants in situ at the time. But at least you can then compare that with the soil modules in Earth system models, where they are imposing a Q10 for sort of similar thing, and that's generally about 2, maybe 1.9. So we see a higher immediate direct response. And then the fall warming resulted in this large burst of CO2 and methane gases that we think was trapped in the talc. And finally, sort of as a conclusion, I think something we need to learn more about is both just how the gradual warming, but also heat waves, more rain on snow events will likely lead to larger CO2 and methane emissions from Arctic tundra. And thank goodness LED will be able to model them at the same time. So thank you. Okay, thank you. That's it.
Thank you so much for your presentation.
Yeah. Again, please remember to put questions in the chat. Here is one from Sufei. Have you observed any change of vegetation in the warming experiment?
We didn't do this long enough to see whether the soil, but I will say that GPP seems slightly elevated, as if more nitrogen had become available because of the warming. You know. There was no change, but we only did this for 2 years. So we just wouldn't see it. Yeah. Anything else?
Yeah. Please keep coming with the questions, but I also have one for you concerning future experiments. So have you any thoughts about that? Also including more than just the temperature? Manipulating more than just that in the field?
I'm sure people here have great ideas for more manipulation experiments, but if I could do it again, or build on this, I would put in the little open top chamber on top, so we're really getting below ground and above ground warming, and then have some plots with nitrogen and without to see whether changing that nitrogen cycling really has a big effect on the vegetation. So, thanks.
Yeah, there's one more question here from Bruno Delisle. How the rain on snow will contribute by promoting the flux? By removing the snow, by increasing the heat flux, by bringing energy below? What are the interactions you foresee?
Yes, thanks. That's a good question. I'm hypothesizing here that the effect of rain on snow is to create diffusive channels, or to sort of remove this impermeable barrier and the energy from the water thawing the upper layer that's also preventing gas flux. Because I'm hypothesizing that the greenhouse gas has already been produced and it's waiting in the talc. However, it's quite possible that that warm energy brought from water just raising the temperature is stimulating microbes too. So it's something to look into. Yeah, thanks. Thank you.
Oh, there's one more question, quick one. From Alga, did you measure any changes in hydrological scale alongside warming? How that will interact with sea cycling?
No, so asking about the hydrology is just a great question, and we just weren't able to in our tiny little mesocosm experiment.
Great, thanks a lot.
Yeah, thank you.
Thank you so much. And welcome to Shurpali, who's going to be talking about eddy convergence, greenhouse gas fluxes from grasslands on mineral and drained organic soils in eastern Finland.
Okay. Thank you. All right, I need to stand here. All right, I will be I'm representing Natural Resources Institute in Finland, and I'm going to talk about eddy covariance flux measurements from different types of grasslands in Finland. And okay, talking about Finland, probably Finland is by far the northernmost agricultural country in the world, and Finnish agriculture focuses on grasslands and croplands, and there is about 1 million hectare area under croplands, and about 2/3 of that on grasslands. So such a large area of grasslands, that means that the two sectors of milk production and associated beef production, they are interdependent and they are quite important to the national economy. And looking at the climate action for the nation and bioeconomy strategy, Finland has set very high standards or targets, to be the first carbon neutral country and reduce the greenhouse gas emissions by 2035 or so. So that is why there is a need for understanding where are the sources and sinks of greenhouse gases. And the urgency of the matter is very clear in one of the recently concluded Ministry of Agriculture and Forestry funded project of mine. And not only, of course, the research organizations and the universities were keen on participating, but I got an overwhelming support from the allied industries. That was something nice to know. Okay, for this, to understand the sustainability of these grasslands and the climate impacts, Luque has invested on a very standardized greenhouse gas infrastructure. And here I'm showing the 3 sites which are within about 5-kilometer range from each other. Antila, that is closer to the lake border there, that is on a mineral site. And Särkisuo and Pappilansuo, these 2 sites are the drained peat soils. And Antila, being a mineral soil, supports cultivation of legume grasses, while Särkisuo and Pappilansuo, they do not support legume crop cultivation, and they are with cereal grasses, cultivated with cereal grasses. And we had, we did not have all the instrumentation in the same year, so that is why it took us, there was a staggered process of getting different instruments from different companies, and that took us some time to establish this GHG network. But 2022 was the first year when we had all 3 sites measuring continuous all three greenhouse gases. So I will give you a sort of a glimpse of what kind of greenhouse gas balance we studied, we observed at the 3 sites here. Okay, just a picture of the 2 sites, Antila, then Pappilansuo, and Särkisuo. And please pay particular attention to the fact that the Pappilansuo site is quite poorly drained, and there is a lot of moisture there. And especially 2022 was a very wet year. So what repercussions this sort of climatic conditions and these kind of mineral versus organic arrangement of sites, what did that have? Okay, during the year, here is a list of the different farm management operations. There were 2 sets of fertilization events at each of the sites, and there were 2 cuts in Antila and Särkisuo, and Pappilansuo was treated a bit differently in the sense that there was a summer plowing. Usually in Finland, whenever you want to finish grassland rotation, you plow the field in the autumn and then keep it ready, and the site is without any vegetation, or the winter, and then next in June, the grasses are again, the site is tilled again, and the grass is planted. But for Pappilansuo, this one of the organic sites, we wanted to try a different tillage treatment, and we wanted to take the first harvest, and then after the first harvest, we decided to plow the field, because this seems like is a preferable management practice the farmers would like to follow. The idea here is that over the entire winter period, we will keep the vegetation on the ground, And after the first cut, we harvest the site and then do a fresh plantation, and with temperatures and moisture conditions being conducive, the growth will happen, and soon we will have again harvestable biomass ready on the field. Okay, but did that happen? Let's see. Okay, here what I'm showing is the net ecosystem exchange for all the 3 sites for the year 2022. And what you see there is as soon as the temperatures start to warm up in the middle of May or so, the vegetation starts to take up CO2 from the atmosphere and reaches a peak. And when the biomass is at its peak vegetative growth, the grass cut is made. And as soon as the grass is cut, you can realize that we are taking out actively photosynthesizing part of the system. And then you see the system turns to a ecosystem source. And with the conditions being favorable for the regrowth, then you see again negative sink. And this pattern was similar for both Sarkisso and Antila. However, what we see for Papilansuo is something very different. As soon as the grass was cut in late June or so, as I said, we performed a summer plowing, and the idea here was to start the regrowth process as soon as possible. But what we realized was it took nearly 7 to 8 weeks for the vegetation to, really the ecosystem to restore its sink capacity. And here is the cumulative net ecosystem exchange. Antila site, being a mineral site, was close to about 800 grams of CO2 flux. On the other hand, the Papilansuo site, which had the summer plowing as one of its management operations, was already at this stage it was a significant source of CO2. And Sarkisso, it was somewhere in between. And we also had methane flux measurements, and here are the cumulative methane emissions. And Sarkisso especially was quite wet and released a lot of methane to the atmosphere. And that the rates of release of methane, they are more or less the same as a natural peatland. And Antila, on the other hand, being a mineral soil site, had a very low methane emissions. And we also had nitrous oxide fluxes. Antila site, which was cultivated with a with a legume grassland. Legume grasslands take up nitrogen from the atmosphere, fix the nitrogen, so they have a lower fertilizer requirement. And the emissions from this site, they are quite low. And then Sarkisso, which also received 2 doses of fertilizer application, was somewhere in the middle. On the other hand, what was interesting to see was that Papilansuo released about 3 grams of N2O per meter square. That is about 30 kilograms of N2O per hectare. That's a huge flux. So we have now calculated the greenhouse gas balance from each of these sites for the 3 sites. And CO2, Antila was close to 9 tons of CO2. Sarkisso 3.5. However, Papilansuo released already nearly 2.6 tons of CO2. Methane, on the other hand, Sarkisso was the significant emitter, and surprising element was nitrous oxide, which released about 9 tons of CO2 equivalent. And if you look at the total greenhouse gas balance, Papulansuo amounted to about 13 tons of CO2 equivalent. So this raises a very significant question. Should we continue to use these organic soils for agricultural purposes? That's an important question, but we cannot just tell the farmer that, sorry, numbers look like this, please stop cultivating. So there are social and cultural and economic issues associated with it. But at least these data will will let the government make certain policies regarding this. And some important points to remember here. Spring phenology. While the two organic soils responded to changing climate, changing climatic conditions during the season, they had a snow melt already in the middle of April or so. However, Antilla site took some time. The mineral site took some time in having complete snow melt at the site. But nevertheless, the very next day, the snow melted from the mineral site. GPP started. GPP was greater than 0 at the Antilla site. On the other hand, for these two organic soils, it took nearly 14 to 20 days nearly to regain the carbon sequestration activity. And as we saw, grassland management practices have a lot of influence, and whether a site is a greenhouse gas sink or a source depends upon what kind of management practices we have there, and this emphasizes these kinds of data emphasize the need for continuous monitoring of greenhouse gases. And also, these data show how we can manage nitrogen management at these agricultural sites. And one other thing that I would like to highlight here is that these organic soils, they have their soil hydrology plays a major role in the overall greenhouse gas balance. And the last slide here is, having studied these two types of organic soils here, one Sarkisuo, which was relatively well drained, I would say, and compared to that, the Papulansuo site was very poorly drained site. And after the summer plowing, despite there was a regrowth of the grassland on the Papulansuo site, we couldn't even harvest the crop because there was so very little because of the wet conditions, and the instruments could not be worked on the field. So there was only a single harvest. So it seems we got to thinking about this, and then it seems that there are a lot of farmers who use these kinds of poorly drained organic soils and just take one cut per year. I mean, that's a very, very environmentally unsafe method. So we came up with an idea with Finnish Geospatial Research Institute. It seems with their satellite images, they can detect which of the sites, organic sites, can have two cuts and which of the sites has a single cut. So with our emission factors, we are now working on this article here and try to sort of find out what percentage of organic soils in this region are poorly drained and what percentage, well drained. Okay, I think I will stop here. Thank you.
Thank you so much. That was very interesting. So there is a comment from Timo Vessel as more comment than a question. He just says that we shouldn't take the current Finnish government's he says, don't take too seriously Finland's sea neutral target because the present right-wing government tries its best not to reach these targets. Yeah.
That's true, yeah.
Unfortunately, yeah. So please just put in comments. I have a question. So in Finland, how large a percentage of the agricultural land is drained peatland, and how big percentage is the mineral soils of the agricultural land?
Well, when you look at southern part of Finland, there we have I don't right now off of my head, I don't know the exact percentages of mineral versus organic soils, but the proportion is quite high in the southern part. Okay, the proportion is rather quite high in the southern Finland. In the northern Finland, mostly it is organic soils.
Thanks. And just to follow up on the nitrogen management of the farms, would it make sense to reduce the nitrogen inputs to the organic soils, you think?
This was the question I was talking to our agronomist in our institute. Well, even on the mineral side, Antila side, what we have seen, we have now from 5 to 6 years of N2O flux data from that side. What we have seen is after cutting the grassland, if you right away apply the fertilizer, it doesn't help much because there is no vegetation which is actively photosynthesizing and taking up that soil nitrogen. So quite a bit of it is released as an episodic release of N2O. So we did an experiment on that mineral side, and last year we waited about 10 days after the first cut. And what we have seen is we have significantly reduced that episodic release. So you are right, we should, especially Pappilan site, the organic site, there soon after the harvest, there was probably no reason to right away apply 80 kilos of nitrogen there. So yeah, even there, if we had waited a little bit, but the expectation was that the vegetation being, the climatic conditions being favorable for a good regrowth right away, that was the expectation. So this is again another take-home message for those farmers who are thinking of doing this summer plowing.
Great, thanks a lot.
Thank you.
And now we reach the last presentation. Yun Bin, who's going to be talking about sustainable use of peatlands for agriculture in the Arctic.
Thank you so much, everybody. And it's almost the end of the day. I hope you're still not too tired. So I'm going to talk about agriculture again, but in Arctic Norway. And it probably seems like not so many people can connect the Arctic to doing agriculture. It feels like a dumb idea to do agriculture in the Arctic. But actually, this in Norway, this big area, and a lot of people are actually living on agriculture in Arctic Circle. So this is some of the situation there. So the government actually promote or encourage the people to move up there and develop the land to do agriculture. But at the same time, those agricultural land you cannot grow like normal crops. So most likely it's like in the situation in Finland, people are growing these fodder grasses and to feed the livestock there. Why do we use peatland in the Arctic then? If we look at the map of Norway, and the peatland actually account for almost 10% of the land, and in the Arctic, and if you look at the distribution, it's basically everywhere. So if people want to use the land there, it's really hard to avoid using peatland up there. And the peatland has a big problem now because once people want to use the land, first they need to drain the land in order to have a lower water level, and because most of the crops doesn't tolerate the high water level. And this created this big problem of CO2 peat decomposition, which produces a lot of CO2. And this is, I think it's a global issue everywhere. And there's a lot of people talking about how to solve this problem. And here are a few numbers in Norway, so you get to know how serious the problem is. And they're up in Norway. And for the drained peatland used for agriculture, it only counts for a very small fraction of the land. But at the end, according to last year's national accounting, it's around 7% of the emissions are actually from these areas. It's like these small hotspots are kind of burning peat all the time, so it emits a lot of greenhouse gases throughout the year. And there are a lot of solutions people are talking about, like rewetting and or do culture, so the farmers can have some income. But all of this, we have to carry out some kind of experiment and measure the actual greenhouse gases coming out from those lands, right? And so for our site, it's located very up north, and we have a institute station up there, and it's at the border between Russia, Finland, and Norway. And the site, we have this drained peatland, and it's cultivated with grass. And we use automatic chambers to do the measurements. Of course, this is an off-grid site, so we have to run this on solar panels, and also we have a diesel generator which produces a lot of power as well. We have 5 plots, and this is how give you an impression how the chambers work, because I think a chamber is definitely a very effective way that we can investigate at a plot scale, and we can input some treatments there. So the chamber works automatically. It opens and closes at the top, and it's just controlling the lead. So most of the body of the chamber is just covering on the ground. And then at the site, we have 5 plots that represent 5 different water levels. And we have 2 fertilization treatment. One represents what the locals do, and one we just double the dosage. And we have 30 of those automatic chambers. So we use Picaro that measures CO2, methane, and N2O gases. And in the first season, we harvest once, and in the second season, we harvest twice. So here is how the climate condition look like during the growing season. So throughout the growing season, the average air temperature is around 9.6 degree, which is not too bad for Arctic site, right? And for the water levels, we have a really great contrast from the plot 1 to plot 5. We have almost 60 centimeters of difference. So it can well represent the wet site, or we consider it as a rewetted site, and well-drained site. And as you expected, the automatic chambers produce a lot of data, and we can see some patterns here, and the harvest definitely play a big role here. But if we combine all those data and calculate the budget, and we see a very nice pattern that in the dry site, and it's still a carbon source, but when it go to the west side, it already turned into a carbon sink. This is quite promising, and for the first year, we harvest only once. And for the second year, we harvest twice, so that somehow make the emissions a bit more over the season, but still, in the west side is still kind of carbon neutral. And we did some photosynthesis measurements this season. It seems those plants in the west side is somehow physiologically stressed a little bit if we compare to the other sides. So that means the decrease of emissions are not actually contributed to those photosynthesis from plants. And if we look at the methane fluxes, it's as a way it's not very surprising that we can see that in the dry side there's a small sink, but in the wet side it's already turned into a big source. It's actually not that big. And the N2O fluxes, the pattern is not that easy to identify. But if we look at the seasonal pattern, I think this is a really interesting pattern. And after the first fertilization, we see just a big boost of N2O emissions. But the second fertilization doesn't have such kind of effect. And I think this is probably due to the vegetation growing throughout the season. And at the end of the season, if we can see, oh, sorry, can I come back? Oh, yeah. And for the end of the season, as Margarita also showed, like those shoulder seasons, it's very interesting. We have also detected this big boost of N2O emissions. But if you compare with temperature, we can see during that period, actually, those the land start to freeze. So if we know some of the, have some experience for chamber measurement, we know during the time when the land starts to freeze and the chamber is actually acting as a warming device. So it's actually like a chimney. All the gases from below ground will somehow goes through these chambers. So I think this kind of fluxes has some artifacts, artificial effects. But I think the most interesting part of this is that if we can see the colors, it's mostly red colors. That means most of the flux are from the dry side and not from the wet side. And I don't know how to explain that, but it's probably worth some further investigation. And for the seasonal greenhouse gas budget, we usually use this global warming potential to represent that. And if you remember the pattern of the CO2, and this pattern is pretty much mimic the CO2 emissions. That means at our specific site, methane and N2O are not that important. They are just very marginal compared to the CO2 emissions. But the story doesn't stop there because, as I mentioned, it's a managed site. So we need to harvest those biomass at the end of season or during the growing season. So we can see a very general pattern that in the dry site, the biomass is much more than the wet site. That also explains why people want to drain the peatland because they produce more biomass. But if we convert this biomass into carbon, then we will see this pattern that all of the plots, no matter what this water table look like, they will turn into a carbon source, just as what Shupali has presented here. And so if we think of this negative, if we think of all of the plots are actually carbon source, they're degrading over long term. So when this come back to the title of my talk, is this kind of management sustainable for using the peatland there? And apparently the answer is no. And of course, there are probably other ways we can improve. For example, if we can change the crops there, maybe it will produce more biomass, or we can change the scheme of harvest. Maybe it could emit some, somehow mitigate some of the carbon loss. So my main takeaways is that rewetting is definitely working and can reduced quite a bit of CO2 emissions. And then at our specific site, N2O and methane emissions are marginal compared to CO2 emissions. And N2O budget are mostly contributed by those hot moments, which we need to do more investigations to understand the mechanisms behind that. And the harvest, of course, is a very, very important factor that we need to consider, and it definitely controls the ultimate carbon balance of the site. And last but not least, you're welcome to visit our site. I think it's a very unique site up in the Arctic, and we have a very nice gradient of water levels. It's like a very good playground for manipulation experiments. So welcome, and welcome. Any kind of collaboration will be welcome. Thank you.
Thank you so much. Are there any questions? Come here. Yeah, come here. Now we do it old school.
Yes. Thank you. I was wondering if you could compare the results you saw for harvest, and maybe a single harvest being more sustainable or less impactful on the carbon compared to the previous talk. Thanks.
Yeah, that's I think how many cuts we need is definitely a very tricky question. And according to what we have found here, and if we apply 2 cuts, then definitely it will lose more carbon. At the same time, probably it will enhance some of the photosynthesis activities after the cutting.
Clearly, there is a need for us to talk to each other.
Yeah, sure.
There is a question now from the chat. It's from Olga. Did you observe structural changes in vegetation community alongside functional differences in photosynthesis, for example?
So the photosynthesis actually we just carried out once, so we cannot really draw any conclusions based on that. But according to our understanding based on that single campaign, I think the photosynthesis is not that important compared to decomposition.
Great, thanks. Yeah. Oh, we have a question.
Oh, now we're gonna get.
Are we going to get scolded? Yeah. Okay. You're going to get scolded, sorry. Well, you can type it down, but otherwise, or you can ask it afterwards. I would like to thank you for participating. Thank you for the presenters. And I hope also to see you tomorrow at our session as well. And the posters as well. Yeah, and have a very nice afternoon or evening.